Hello, and I warmly welcome you to what's my pre-recorded video of my OHBM presentation. In this short talk, I hope to discuss some of the I find exciting approaches where we utilize the understanding of the physics and physiology of the head and the brain to disentangle the signals of interest that are coming from the cortex from noise and other disturbances. And this can be very valuable in the field of TMS-EEG data analysis. In the latter part of the talk, I want to touch upon some of the pressing matters that we have found recently related to the replicability of different TMS-EEG pre-processing pipelines. And I plainly want to open the discussion for how could we get into the root of this problem and how we could in the future find what is really going on and improve the replicability. But without further ado, let's get started. We have a lot of things to go through together. So, of course, the idea of combining transcranial magnetic stimulation with electroencephalography would be to measure directly the responses of the cortex when we stimulate it with the transcranial magnetic stimulation. Unfortunately, the strong magnetic pulses also easily create many sorts of artifacts and noise signals that we would want to remove before we start to analyze the interesting good stuff that is the TMS-EEG of the brain. Luckily, there are some aspects of the physics and physiology that actually end up helping us to do this task to remove the noise. Namely, we know very well that the neuronal EEG signals are coming from the post-synaptic currents under the skull. And because of the conductive properties of the skull and the scalp and the kind of electromagnetic properties of the post-synaptic currents, we know that these brain sources produce voltage patterns on the scalp that are rather smeared and spread out. And this can be very different from noise and artifact signals, as we will see just in a minute. So first I will introduce you the so-called signal space projection combined with source-informed reconstruction method that we developed some years ago for removing the TMS-revoked muscle artifacts. And as you might know, when we stimulate different parts of the brain, we can easily create some muscle responses that we are not interested in and that can easily mask the neuronal signals. We can see here that those responses are especially bad on the lateral parts of the head, and also from this traffic lights image, we can see that whenever we stimulate regions closer to the muscles, the problems get elevated. Whereas in the vertex, the situation is much better. We can also on the left see that the muscle artifacts have this characteristic biphasic response, and the topography is coming from the lateral part of the head. But we also know that if we use our forward modeling techniques and try to model the cortical signals that we would expect from the, for instance, from the primary motor cortex, even with the different types of conductivities and what have you, we can see that these topographies would be rather smeared and spread out, very different from these what we observe when we have a big muscle artifact. So the question is then, how could we remove those muscle artifact responses? And again, we use the physics and physiology because the muscles are superficial and they are lying on the skull. We know that the frequency band at which their spectrum is observed in the EEG is much broader than the frequency spectrum of the neuronal signals that we know that are expected to be below 100 Hz. So if we look into the high pass filter data, we can use that to estimate the topographies of the artifacts that we can then project out with the signal space projection method. This was already tested by Mäki and Ilmoniemi in 2011. But the problem is that although it is it is removing the muscle artifact, it is also clearly attenuating the signals of interest. And we decided to test an approach which we call the source-informed reconstruction, where we actually use our forward model of the head to reconstruct these clean signals in the original signal space, so that we could at least minimize the unnecessary attenuation in the signals of interest. And the idea is seen in this figure, which is rather simple. Sorry. So we first project out the artifact topographies, and we then use this information that we have left to calculate the inverse estimates of the cortical signals, artifact-free cortical signals, which we can then use to forward map using our forward model to the original signal space. And this seems to work quite well. In the end, we have a very simple spatial filter that defines our clean signals in terms of the original signals. Although you can see here the logic that led to this very simple equation. Now, here you can see some TMS EEG responses that we have cleaned from these muscle artifacts. The red ones are the original signal and the black ones are after cleaning. You can clearly see that under these very high frequency topographies, we can actually undercover topographies that are much more spread out and actually do correspond to the topographies that you would expect from the primary motor cortex if we do some further forward modeling. You don't only have to restrict the SSP SIR response to muscle artifacts, you can use it to other problems that are similar. And with Piabani et al, we tested whether we could use a similar approach to remove the somatosensory responses that are related to the TMS stimulation. As this is a problem that was, of course, revealed by Conde et al in their famous paper some years ago. And here we had a control measurement where we actually stimulated the shoulder with the TMS. And by measuring the topographies from that control data, we then use the SSP SIR to project out those topographies from the TMS EEG responses and then reconstructed those signals. You can see that the curves here are quite attenuated. But this is to be expected because both of these responses are now coming from the cortex. However, if we look at the topographies, we see something quite interesting. We see that we can actually recover a quite different looking topographical patterns, especially in the beginning. And it gets even more interesting if you look into the source estimates after this cleaning. So without cleaning, both the direct stimulation of M1 with TMS and the shoulder stimulation actually result in surprisingly similar looking brain responses. But after cleaning, we can actually recover a very different looking pattern that is much more emphasized on the primary or on the motor system. So that was interesting and kind of looking potential, showing potential for this method. The other method that we developed a couple of years ago that uses very similar idea is the so-called sound algorithm. We are actually pretty excited about. The idea is pretty simple. So we cross-validate all of our channels first by using this iterative inverse and forward computations, and we compare what kind of predictions we would get for each channel from the other channels and compare them to the measurements. And using this iterative scheme, we can actually find a spatial filter that removes non-correlated noise from the TMS EEG responses. And here you can see some example data, so example TMS EEG data, how well this algorithm is able to find these contaminated channels automatically and replace them with the best possible prediction from the other channels. And the cool thing about the sound approach is that, again, because it is such a simple spatial filter in the end, you can actually quite easily take this approach and try to use it in different applications. And we have recent years, we have been testing this method for the real-time cleaning purpose. So the idea is that in a parallel process, we read data to a buffer, which we use then to estimate this noise cancellation, spatial filter, sound filter. And whenever the update is ready, we can then fast apply the spatial filter to the streaming data, resulting in nice looking clean data. And the nice thing about this approach is that whenever there are new bad channels introduced, for instance, during a long, let's say, closed loop EEG TMS experiment or EEG informed TMS experiment, we could react to that. And here is some first results that we have obtained. You can see that it quite nicely finds some of these contaminated channels over here, AF4, AF3, and also this, if there are blinks, those can be removed with a kind of a SSP type of approach. So we think that this is very promising. The other nice aspect about this spatial filter forward model-based spatial filtering methods is that because they are just linear operators, it is very easy to quantify the overcorrection. So once we have calculated these spatial filters from our data, we can then take our forward models and apply them and ask the question that, okay, if this red arrow shows our stimulated location and that produces our data that further produces our spatial filter, how would different possible sources in the cortex then be attenuated? And in this specific example, we can see that, for instance, here, removing the muscle artifacts would indeed attenuate the cortical sources close to the coil. But if we are interested in cortical connections, for instance, on the contralateral hemisphere, we can quite safely measure those responses even after cleaning. So that's very nice. These methods allow you to quantify the level of overcorrection. And now finally, I will spend a couple of minutes talking about the recent findings that we have got from comparing the different pipelines for DMS EEG pre-processing. So as you might know, there are multiple different pipelines, different groups are using different combinations of independent component analysis, filtering, all these forward modeling based methods that we described. And very recently, Bertazzoli et al., we wanted to compare how similar results would we get from different popular pipelines. And unfortunately, the result can be summarized that there were quite big differences in the topographies, but also in the overall curves. And then in the next project with Nigel Rokas et al., we then kind of wanted to expand a little bit on this question and only concentrated on a very specific step. So we just removed a decay artifact with 3 different methods. So fast IC, a sound, and a fitting a model. And we just wanted to remove, like, okay, or remove this decay artifact and just ask the question, okay, how well could we get corresponding results from this very simple step? And actually, even there, we did find some differences. So this suggests me that, you know, there are differences in the pipelines, and the reasons for these differences can be quite complicated. So it can go down to the single step level, and especially it might be very critical how we combine different steps. So how do we combine filtering, high pass filtering? Should it come before or after ICA? What about different drifts and so forth? I think this is a problem that we need to tackle in the future as a TMS EEG community. The problem is that we don't have any ground truth. And so in the future, we need to come up with some sort of wise experimental methods, like, for instance, combining event-related potentials that don't have any TMS, and we know those responses, and then add some experimental noise or signals that mimic muscle artifacts and so forth, so that we could evaluate different pre-processing methods step by step tediously going into details to find out what is really going on in this non-correspondence issue. So as conclusions, I hope that this very brief run through of this recent works that I have been involved has convinced you that these forward modeling based methods can be highly valuable in detecting and removing different artifacts and noise signals. They have some very nice merits, like they allow a straightforward quantification of overcorrection, and they can be used to automatically detect and reject noise components even in real-time applications in the future, which is pretty cool. However, unfortunately, there are some aspects in our TMS-ET field that we do need to solve in the future. That is, different pre-processing pipelines seem to cause different outcomes. I'm afraid that the devil is probably on the details, and that's why we would really need innovative experimental approaches and step-by-step validations in the future to find out what is really going on here. And with this, I want to thank all of my collaborators that have contributed to these works or work over the previous years, and also, of course, the funding agencies who have always supported me to do this work. Thank you very much.